Hello my students, how are you all? Welcome back to my EduSpace. Hope you all are doing and learning well. If you like my videos, then don't forget to subscribe my channel and you can share to your friends as well. So let's talk about today's video. It's about hematopoiesis. As I already told you that we are continuing the series of immunology. So we will concentrate more on the formation of WBC or white blood cells. Because they are the part of body's immune system, they help the body to fight against infections or other disease. So let's kick it off. Now, what is hematopoiesis? It takes from the Greek word which means heme and poiesis. Heme means blood and poiesis means formation or to form. So it is the process of formation of blood cellular components which are derived from the uncommitted hematopoietic stem cells. So this is the new term. Let's we talk about what is hematopoietic stem cells, or we can say HSCs. Actually, they are present in bone marrow. Suppose this is our bone. Okay. And if we transect this part, we will see that there is an internal core which is made up of spongy tissue. This spongy tissue is known as bone marrow. So they are present in bone marrow, that is spongy part of bone at the core of the bone, we can say. As they are stem cells, they have the ability to give rise to all different types of blood cells. So as they are stem cells, they can be changed or differentiate into any type of cells. And if we are talking about the hematopoietic cell, so it means they can be changed into any other type of cells that are present in our blood. They are self-renewing. It means the hematopoietic cells are self-renewing, that they are not completely depleted during differentiation, but some of these daughter cells remain as hematopoietic stem cells. So suppose this is hematopoietic stem cells. When it's getting differentiated into daughter cell, some of the cell goes to different type of cell lineage, but some of its daughter cell remain as like the same. It means they retain its originality. So they are self-renewing. They are not completely depleted during differentiation, because and we can say that some of these daughter cells will remain as hematopoietic stem cells. The process of hematopoiesis begins during embryonic development and it continues throughout the life to produce and replenish the blood system. So this process has started during the embryonic development and it continues throughout the life. There is a formation of approximately 10 to the power 11 to 10 to the power 12 new blood cells per day. Such a big, huge amount to maintain the steady state levels in the peripheral circulatory system. So hematopoiesis plays an important role for replenish the blood system. Now let's learn about how the hematopoietic stem cell differentiated into a particular type of cell lineage. So first, the hematopoietic stem cell can be divided into two pathways. The first one is our myeloid progenitor cell and the second one is lymphoid progenitor cell. So we can see that hematopoietic stem cells can be differentiated into either one of the two pathways, and these two pathways are myeloid progenitor cells and the lymphoid progenitor cells. The progenitor cells lost the capacity of self-renewal and are committed to particular cell lineage. So as this hematopoietic cell that is self-renewal, okay, but as soon as it committed to a particular type of progenitor cell, then it lost the capacity of self-renewing. Okay, as this hematopoietic cell is self-renewable, but the myeloid progenitor cell and the lymphoid progenitor cells, they are not self-renewing. Okay, next. So as we just see that the hematopoietic stem cell is differentiated either into two pathways or two progenitor cells. The first one is myeloid progenitor cell and the second one is lymphoid progenitor cell. So we will look in this particular slide about the myeloid progenitor cells, how these myeloid progenitor cells will get differentiated into different types of cell lineages. Okay, suppose this is our myeloid progenitor cell. It can be differentiated or the progenies formation of, or it can act as a progenitors of RBC, okay, or we can say erythrocytes, and this process is known as erythropoiesis, where the formation of RBCs takes place. So this process is known as erythropoiesis. RBC poiesis formation. So in this, the myeloid progenitor cell give rise to the cell lineages or the formation of RBC, and this process is known as erythropoiesis. Okay, and it can also form the granulocytic WBC. Granulocytic WBC, it reflects the neutrophils, basophils and eosinophils and also the formation of monocytes. So in this category includes the leukopoiesis, because here is the formation of WBCs takes place, so it is known as leukopoiesis. Okay, and also myeloid progenitor cells can be differentiated into megakaryocytes, which ultimately leads to the formation of platelets. Okay, so this process is known as megakaryopoiesis. This process is known as megakaryopoiesis. Megakaryopoiesis. Okay, so the myeloid progenitor cells can do leukopoiesis, erythropoiesis or megakaryopoiesis, and it leads to the formation of red blood cells through the process of erythropoiesis, formation of WBCs and monocytes, and also dendritic cells in the process of leukopoiesis, and also the formation of platelets in the process of megakaryopoiesis. Now, the second pathway of hematopoietic stem cell is lymphoid progenitor cell. So, it can generate or progenitors of lymphocytes and natural killer cells. It can be differentiated into cell lineages of lymphocytes and natural killer cells. Lymphocytes are B lymphocytes and T lymphocytes. So this is through the lymphoid progenitor cells, there is a formation of B lymphocytes and T lymphocytes and also the formation of NK cell. The formation of B and T lymphocytes also comes under the process of leukopoiesis. So we will more concentrated on the leukopoiesis as we are taking the immune system here. The immune system, the cells that are responsible for generating an immune system are basically WBCs. Okay, now the role of stromal cells. Stromal cells includes fat cells, endothelial cells, fibroblasts, and macrophages. Basically, they influences the differentiation of hematopoietic stem cells by providing hematopoietic inducing microenvironment. So, how this hematopoietic stem cell knows that they have to differentiate into a particular type of cell lineages. So, this type of we can say the connection or call is given by these stromal cells. These stromal cell influences the differentiation of HSCs by providing the hematopoietic inducing microenvironment. This microenvironment consisting of cellular matrix and the factors that promote the growth and differentiation of hematopoietic stem cells. So these stromal cells are responsible for the factors the factors that promote the growth and differentiation of hematopoietic stem cells. So these factors may be soluble or may be membrane bound molecules which are present on the stromal cell. So the hematopoietic growth factors can be a soluble agents that arrive at the HSCs by diffusion. So the soluble particles that are present at the surrounding they arrive at the hematopoietic stem cells through direct diffusions, and these are basically chemicals, or we can say cytokines. Okay, they are basically cytokines or hormones that are responsible for giving signals to the hematopoietic stem cells to get differentiated, and the others are membrane bound molecules that are present on the stromal cell. So suppose this is the membrane bound factors. So the membrane bound factor, this is communicate or get signal to the hematopoietic stem cells, which ultimately leads to the stimulation of the hematopoietic stem cells to get differentiated. So it acts as a stimulation. Okay, so this receptor which is present on the hematopoietic stem cell is recognized this particular factor, which ultimately leads to the activation of hematopoietic stem cell to get differentiated into a particular type of cell lineage. Okay, so these stromal cells are very important or play a very important role in the differentiation of the hematopoietic stem cells. Through this, the hematopoietic stem cells get to know to which they have to get differentiated into what kind of cell lineage. So let's we take first the myeloid progenitor cells. How these myeloid progenitor cells forms different types of cells. So first we are taking formation of monocytes. So this is our myeloid cell, or we can say myeloid progenitor cells. Both are same. You can say myeloid stem cells or myeloid progenitor cells. We all know that the myeloid stem cell is get differentiated into either monocytes or the granulocytes. So they can be differentiated into two cells. The first one is monocytes and the other one is granulocytes. In granulocytes, there is a formation of basophil, eosinophils and neutrophils. So we will see this particular process in the next slide. First, we will concentrate on the formation of monocytes. With the influence of interleukin 3, interleukin 5 and a granulocyte colony stimulating factor, the myeloid stem cell is get differentiated into monoblast. Okay. Now this monoblast is again changed to promonocytes. Why promonocyte is called? Because it is this time it is non-functional. The pro suffix is always used to the primitive version of any kind of cell. So we are saying that this is promonocytes. Here the nucleus is slightly indented or slightly U-shaped. The notch is not that prominent that is present in the case of monocytes. So further, this promonocyte is differentiated into monocytes. So this one is functional and this is present in the blood. So this monocyte is present in the blood and this is very much functional. And when this monocyte is going to the tissue, okay, then it get differentiated from monocyte to the macrophage. If it present in the tissue, it converted or changed its form and become macrophages. Macrophages, as its name indicate, that these are very good in phagocytosis process and these are very big and huge cells, so we can say that these are macrophages. So in tissue, the monocytes are getting converted into macrophages. And if they are present in the CNS, central nervous system, or in the brain cells, they are changed in the glial cell. And in the interstitial spaces, they are changed into the dendritic cell. So, let's take a review. Let's recall it again. The myeloid stem cell is get differentiated either into monocytes or in the granulocytes. In the formation of monocytes, first the myeloid progenitor cell is get differentiated into monoblast and then to the promonocyte. In the promonocyte, this is the primitive cell and it's not functional at this time. The nucleus is slightly notched and this particular promonocyte is differentiated into monocytes where the nucleus is very much of U-shaped or very much notched, and this monocyte is functional in the blood. When from the blood it comes to the tissue, then it becomes macrophage, and when it comes to the brain it becomes glial cells, and in the interstitial spaces it becomes dendritic cell. So our next slide we will gonna learn about how myeloid progenitor cell is get differentiated into the granulocytes, that is basophil, eosinophils and neutrophils. Now the formation of granulocytes. So we have just seen that from the hematopoietic stem cells, two pathways has generated. The first one is myeloid and the other one is lymphoid. From the myeloid, we have seen that monocytes has formed, and in this slide, we are going to look how the granulocytes are formed. So first, in myeloid stem cells, with the influence of interleukin 3, interleukin 5 and granulocyte colony stimulating factors. With the influence of these factors, the myeloid stem cell, or we can say the myeloid progenitor cell, get differentiated into myeloblast. Okay, then this myeloblast generates three types of promyelocytes. The first one is basophilic promyelocytes, the second one is eosinophilic promyelocyte, and the third one is neutrophilic promyelocyte. So we can see from the myeloblast there is a generation of promyelocytes. Okay. Now, this cell is blue in color. Why this is blue in color? Because the granules that are present in the basophilic promyelocytes stained the methylene blue. That's why they appears blue in color. Whereas in eosinophilic, the granules stain red. Thereby, these are red in color. Whereas in neutrophilic, the cell looks quite midway between these two blue and red color. That's why it appears quite pinkish in color, because the granules that are present in the neutrophilic promyelocytes can attain, some granulocyte can attain blue color, whereas others attain red color. That's why the color is quite hybrid form of. Okay. Now, from this promyelocytes, the other type of cell, that is myelocytes, has differentiated. The promyelocytes and the myelocytes are quite similar, but the main difference is that promyelocytes is that cell lineage that formed before the myelocytes. Okay, so this is, as I just told you, pro means primitive. So these are old old cells, and the myelocytes are newer one cells or some advanced cells. In this, we can see that there is a nucleus is having slight notch. Okay, this is not very constricted notch is present, but they are slightly notched in all the cases. Same in the eosinophilic myelocytes and also in the neutrophilic myelocytes. Now, from the myelocyte, they have perfectly constricted nucleus. Okay, and the cells that are having perfectly constricted nucleus or perfectly U-shaped nucleus are known as band cells. Okay, so now from the basophilic myelocyte, basophilic band cells forms, and eosinophilic myelocytes generates the eosinophilic band cell, and from the neutrophilic myelocytes, neutrophilic band cells are formed. Okay, now from this band cell, there is again changes in the structure of the nucleus, and now they are taking their complete form. They form the multi-lobed nuclei. In the case of basophil, it is the U shape changed into S shape nucleus. Okay, and in the eosinophilic, from the eosinophilic band cell, there is a change from the U shape to the bilobed nucleus. This nucleus is bilobed here. And from the neutrophilic band cell, it's having pink color, as I just told you, and it's multi-lobed nucleus. Okay, it's multi-lobed. So this is the formation of granulocytes. It also comes under the process of leukopoiesis. Okay. Now, the formation of platelets. Basically, the formation of platelets comes under the process of megakaryopoiesis. In this, the myeloid stem cell or myeloid progenitor cell under the influence of thrombopoietin. Under the influence of thrombopoietin, this is the hormone that secretes by the liver and kidney. Okay. Under the influence of thrombopoietin, the myeloid stem cells differentiated into megakaryoblast. And this megakaryoblast is, from this megakaryoblast, the pro-megakaryocyte has formed. And from the pro-megakaryocytes, the megakaryocyte is formed. And from this, there is a multiple duplication of DNA inside the nucleus takes place. But in this case, there is no cytoplasmic division occurs. That's why this cell is having such a huge size. That's why it's called megakaryocyte. Okay, it's having big size. One thing you have to note that from this megakaryocyte is so much large that it cannot pass through the sinusoidal capillaries of blood. Okay, these are the sinusoidal capillaries of blood. So for what happened with this megakaryocytes, when it has to cross this, it's get fragmented out, or we can say burst out. So these are the cytoplasmic fragments of megakaryocyte. So it's get fragmented out and form the platelet form. Okay, these platelets are nothing but these are the cytoplasmic fragments of megakaryocytes. So this is the formation of platelets from the myeloid stem cells. The megakaryoblast is formed. From the megakaryoblast, promegakaryocyte is formed, and from the promegakaryocytes, megakaryocyte is formed. Here, there is a duplication of DNA takes place so many times but there is no cytoplasmic division takes place. That's why this type of cell attain a very large or huge size. When they have to cross the sinusoidal capillaries, they burst out into smaller cytoplasmic fragments, and these smaller cytoplasmic fragments are known as platelets. Now, formation of lymphocytes. Till now, we have seen that from the myeloid stem cells, what type of cells are formed. Now, in this slide, we are going to look at the lymphocyte stem cells, how and what type of cells are generated from the lymphoid stem cells or lymphoid progenitor cells. So under the influence of interleukin 3, interleukin 5 and A granulocytes, colony stimulating factors, this lymphoid stem cells is transformed or changed to lymphoblast. One thing I have to say that from the lymphoid stem cells, there are other two types of cells are formed. These are natural killer cells and the dendritic cells. Okay, beside lymphocytes. So it can generate three types of cells: the NK cells, the dendritic cells, and the lymphocytes. So let's continue how lymphocytes are formed. So from the lymphoid stem cells, lymphoblast is formed, and from the lymphoblast, prolymphocyte is formed. Now, from the prolymphocytes, lymphocyte cell, lymphocyte cell is formed. This lymphocytes leaves the bone marrow and goes to the blood and then resides in their lymphoid organs where they get mature. Okay. So, two types of lymphocytes are formed. First one is B lymphocytes, progenitor, which forms B cells, and the other is T lymphocytes, progenitor, where they form the cytotoxic T cell and the helper T cell. One thing you have to notice that the B cells is activated in this phase, but for the T cell, T lymphocytes are still not activated. It get mature. First, it leaves to the thymus where it goes to mature and then become functional. So, it leaves to the thymus. It goes to the thymus, and inside the thymus, the T lymphocytes get mature and then they become functional. So, this is the formation of lymphocytes. Okay, that's all for today. That's all for today, and if you have any query, please text me below and share your suggestions about the topics you want me to cover in upcoming videos. Thanks for watching.